Now, moving on to our next presentation by Hosna Hajati from Department of Animal Sciences, Research and Education Center for Agriculture and Natural Resources, East Azerbaijan, Tabriz, Iran, presenting on the title Effects of In Ovo Injection of Spirulina platanensis, Hatchability and Growth Performance of Broilers and Japanese Quails. Doctor, the stage is yours now.
Thank you so much. Hello, everybody. Can you hear me?
Yes, doctor, yes.
Thank you so much. First of all, I would like to thank for the invitation. I'm Dr. Hosna Hajati, Research Assistant Professor at Agricultural Research Center of Tabriz, Iran. And now I will share my screen. Can you see my screen?
Yes, doctor.
Thank you. Today, I would like to share our recent findings about in ovo injection of Spirulina platensis on hatchability and growth performance of broilers and Japanese quails. The outline of my presentation include introduction, material and method section, and conclusion, recommendation, and at the end, acknowledgements. As you know, heat stress is one of the critical events that has a detrimental effect on poultry welfare and performance due to oxidative damage of cells, endocrine disturbance, immunosuppression, and gut disorders. Chicken embryos are susceptible to oxidative stress due to high content of polyunsaturated fatty acids in their tissues. Also, we know that up to 3% of oxygen in electron transport chain can convert to reactive oxygen molecules that can destroy DNA, proteins, and lipids. Also, phagocyte cells produce reactive radicals which can injure body organs while escaping phagosome. So, due to rapid embryonic growth and the fast metabolic turnover of modern chicks, some of the antioxidant substances in the closed environment of the egg can become inadequate during the late phase of incubation. This restriction of nutrient reserves may limit the maximal development and the growth of newly hatched chicks. Surai et al. In 2003 had shown that a balanced antioxidant system during the hatch is a critical factor for hatchlings' viability. However, the bird's eggs have closed environment, as I mentioned, so it seems that in ovo injection of organic antioxidant may help to reduce stress and mortality of the embryos during the late phase of incubation, in addition to promote their immunity system. Sharma and Burmester in 1982 first used in ovo method to vaccinate against Marek disease. Uni and Furkett in 2003, as we know, they are the pioneer scientists in in ovo issue, patented in ovo feeding and stated that this method could beneficially affect hatchability, intestinal mucosa, and body weight at hatch and also in 35 days of age. But researchers have shown that there are some critical factors for in ovo injection in order to get desirable results. For example, we should notice the egg location, shell penetration, the site and time of injection, injectable materials, injection condition, and also the sanitation of egg and hatchery and material that we use. And also the transfer situation is important for in ovo injection. In this slide, you can see the frequency of Innovo vaccination in modern hatcheries around the world. Today, there are many different countries such as United States, Canada, Italy, Germany, Thailand, China, and so on that use Innovo injection for vaccination the poultry. Our research showed that the effects of Innovo injection of various biologically important compounds such as amino acids, carbohydrates, vitamins, inulin, monooligosaccharide, minerals, hormones, and herbal extract, and etc., had been studied on hatchability and growth of chicks post-hatch. For example, in our previous research, we showed that Innovo injection of 4.5 milligram grapeseed hydroalcoholic extract per egg as a herbal extract with high antioxidant activity on 18th day of incubation increased hatchability of COP 500 broiler chickens. So we are searching for, organic materials that has antioxidant characteristics, such as microalgae like spirulina. Spirulina grows naturally in alkaline lakes, but it is commercially produced in large outdoor or greenhouse ponds under controlled conditions. Spirulina platensis and Spirulina maxima are the most common and widely used species. Spirulina strongly induces antioxidant enzyme activity and helps to prevent lipid peroxidation and DNA damage and scavenges free radicals. In this slide, you can see the image of our recent book about Spirulina platensis in poultry nutrition that is publishing by Cambridge Scholars Publishing. And it is available online. Spirulina platensis is a microalgae rich in antioxidant phycochemicals such as phycobiliproteins, phycocyanin, allophycocyanin, beta-carotene, and phenolic compounds. These substances have been proved that have antioxidant, antimicrobial, anti-cancer, and anti-inflammatory activities. For example, in this image, you can see the mechanism that spirulina mechanism against free radicals, and this is adopted from Wu et al. In 2016. In another of our previous research, we have found that Spirulina platensis decreased malondialdate level in the blood of quails under heat stress. Also, it was found that Spirulina platensis improved immune response of broiler chickens. So, the objective of the present study was to determine the effects of Innovo injection of Spirulina platensis on broilers and Japanese quails' hatchability, birth weight, growth performance, heat shock protein 70 gene expression, and gamma interferon gene expression as an immunity cytokine. In this slide, you can see some of our materials. For spirulina preparation, we were cultivated spirulina in modified Zaruk medium, and harvesting was done after 14 days. After harvesting, the microalgae was air dried, grounded, and analyzed by AOAC 2005 procedures. For Innovo injection in experiment 1, a total of 240 fertilized eggs were used, and they were divided into 6 groups: control group without any injection, sham control with the injection of 0.02 cc distilled water, and also 0.75 and 1.5 2.5 or 3.5 milligram spirulina per egg was injected. The injection was done on 15 days of incubation into the center of air sac of the fertile eggs after disinfection with 70% ethyl alcohol. And also we used sterile paraffin and took back the eggs to the incubator. In experiment 2, a total of 192 fertilized broiler breeder eggs were used, and they were divided into 4 groups. Control group without any injection, sham control that was injected by 0.2 cc distilled water, and 25 or 35 milligram spirulina injected per egg. The injection was done on 18 days of incubation into the center of air sac of the fertile eggs. After disinfection with 70% ethyl alcohol, then the eggs were sealed with nail polish and took back to the incubator. Also, we measured hatchability and hatching weight. The hatchability was calculated as the number of hatchlings divided by the number of fertilized eggs multiply 100. And on day 21 of incubation, after dryness of fluffs, the hatchling of each replicate were taken out of the incubator and weighted. I don't know why I cannot change the slide. Okay. Also, we measured the gross performance of the hatchlings. For example, in experiment 1, after weighting the hatchlings, quail chicks were grown in 24 cages with dimensions of 40 multiply 90 multiply 20 centimeter. 8 quail chicks were raised in each cage for 1 week, and the experimental diet was formulated by WUFFDA software, and it is shown in table 1 in the next slide. And the house temperature and the relative humidity was kept around 35 degrees centigrade and 65% respectively. The house lighting program was 23 hours light and 1 hour dark, and the mean feed intake, body weight gain, and feed conversion ratio of the quails in each cage was calculated as daily. In this slide, you can see the ingredients and nutrient composition of the experimental diet. You can see that the main ingredient was corn and soybean meal, and also we use corn gluten, soybean oil, DCP, oyster shell, common salt, and a vitamin and mineral premix, D-L-methionine, L-lysine, and L-threonine to formulate the diet. In experiment 2, broiler chicks were raised in 16 floor pens with wood litter, and 8 broiler chicks were allocated to each pen for 4 weeks. The experimental diet is shown in the next slide, and that was prepared according to RAS 308 nutritional guideline. The initial temperature and relative humidity of broiler's house was 32 degrees centigrade and 65% respectively. Lighting program of the house was 23 hours light and 1 hour dark. Gross performance traits such as body weight gain, feed intake, and feed conversion ratio was calculated for the chicks in each pen daily. In this slide, you can see the ingredients and nutrient composition of broilers' experimental diets. As you can see, The main ingredients also was corn and soybean meal, and we also used corn gluten, soybean oil, BCP, oyster shell, common salt, mineral and vitamin premix, D-L-methionine and L-lysine to calculate and prepare the diet for the chickens. We assessed gene expression by real-time PCR, the expression of heat shock protein 70, glutathione peroxidase, and gamma interferon was assessed in the liver of one-day-old quail hatchlings in experiment 1. And in experiment 2, the heat shock protein 70 gene expression was evaluated in the liver of one-day-old broiler hatchlings. We use GAPDH as an internal control gene, and a real-time PCR system was used to amplify the genes. For evaluating gene relative expression, delta-delta CT model was used in present study. And you can see in the bottom of this slide, the gene forward and reverse primer sequences in experiment 1 and in experiment 2. Also, on hatch day of the experiment 1, the level of MDA, malondialdehyde, and catalase activity in the liver of quail chicks was measured by spectrophotometry. In order to analyze our data, arc-sign transformation was used for the percentage data, and the statistical model in the 2 experiments was a completely randomized design, and all data were analyzed by GLM procedure of SAS software. Duncan's test was used to compare the means of the considered traits, and the level of significance was considered as below 0.05. I will start the results section. The effects of in vivo feeding of Spirulina platensis on hatchability of Japanese quails are shown here. And as you can see, in vivo injection of Spirulina from 1.5 up to 3.5 milligram spirulina per egg has positive effect on the hatchability of Japanese quails. Around 10% improvement we saw. And in this slide, you can see the effects of in vivo feeding of Spirulina platensis on birth weight of Japanese quails. And as you can see, Spirulina platensis improved again the birth weight of Japanese quails. For example, when we compare treatment 6, I mean 3.5 milligram spirulina injection caused around 11 improvement in the birth weight of chicks compared to control group. In this slide, you can see the effects of in vivo feeding of Spirulina platensis on chick body weight of Japanese quails at 2 days of age. And as you can see, in vivo injection of Spirulina platensis from 1.5 to 3.5 milligram per egg had positive effect on the body weight of Japanese quails at 2 days of age. And when we compare treatment for, 1.5 milligram spirulina injection to the egg, improved the body weight of the Japanese quails around 7% at 2 days of age. Here you can see the effects of in vivo feeding of spirulina on chick yolk residue of the Japanese quails at 2 days of age. And as you can see, when we compare treatment 5 with control group, we can see around 30% reduction in chick yolk residue at 2 days of age. And this is desirable for gut health and for improvement, the bird and higher performance in the farm. In this slide, you can see the effects of in vivo feeding of spirulina on chick body weight of Japanese quails at 7 days of age. And when we compare treatment, we found that in vivo injection of distilled water as sham control had negative effect on the chick body weight at 7 days of age. Here you can see the effect of in vivo feeding of spirulina on feed conversion ratio of Japanese quails on 7 days of age. And you can see that in vivo injection of spirulina from 1.5 up to 3.5 milligram per egg had positive effect on FCR, and it reduces the FCR or feed conversion ratio. For example, when we compared treatment 5 with the 2.5 milligram spirulina injection, decreased FCR around 5%, and it is valuable for the farmer. In this slide, you can see the effects of in vivo injection of spirulina on liver catalase activity of quail chicks on hatch day. And we see that in vivo injection of spirulina 2.5 milligram or 3 at the level of 3.5 milligram had positive effect and increased liver catalase activity. For example, when we compare control group with the group that in vivo injected by spirulina around 2.5 milligram, it affects positively on liver catalase and increased the liver catalase activity around 28%. In this slide, you can see the in vivo feeding of spirulina effect on HSP70 or heat shock protein 70, glutathione peroxidase, and gamma interferon gene expression in the liver of 1-day-old quail chicks. And as you can see, in vivo injection of 2.5 or 3.5 milligram spirulina decreased the heat shock protein, and heat shock protein, you know, that is a biomarker of heat stress condition. And also in vivo injection of 2.5 or 3.5 milligram spirulina per egg increased the glutathione peroxidase gene expression in the liver of hatchlings on the day of hatch. And also in vivo injection of 1.5 or 2.5 or 3.5 milligram spirulina per egg increased gamma interferon gene expression. And we know that gamma interferon is a cytokine that is important for immunity system and has positive effect and promotes the immune response of the chicks. Here you can see the effects of in vivo feeding of spirulina 25 or 35 milligram per egg on hatchability of broiler chicks in experiment 2. We didn't see any significant effect. Also, 25 milligram or 35 milligram spirulina injection increased hatchability numerically. Here you can see the effects of in vivo feeding of spirulina in broiler chicks on yolk residue on day of hatch in broilers. And when we compare treatment, we saw that 25 milligram or 35 milligram spirulina injection had positive effect on yolk residue. I mean, the birds that were injected by spirulina had lower yolk sac residue. Here you can see the in vivo feeding of spirulina 25 or 35 milligram per egg on heat shock protein 70 gene expression in the liver of 1-day-old broiler chicks. And as you can see, in vivo injection of spirulina platensis per egg had positive effect on heat shock protein 70. I mean, it decreased the heat shock 70 heat shock protein 70 expression, and this is a biomarker of heat stress. So it shows that spirulina can reduce the heat stress condition for the embryos. Here you can see the effects of in ovo feeding of spirulina in broiler chickens on the performance of broiler chicks up to 4th week of age. And when we compare the performance of the birds, we saw that spirulina injection at the level of 35 milligram per egg increased feed intake and also increased feed conversion ratio of the birds during the 4th weeks of rearing. And so we can recommend 25 milligram spirulina injection is better, and we had seen no difference among feed conversion ratio between 25 milligram in ovo injected birds or control group. In conclusion, spirulina platensis have the potential to be considered as a functional organic supplement in commercial hatcheries. In ovo injection of spirulina platensis can improve hatchability, antioxidant, and immunity-related gene expression in quail chicks. And also, in ovo injection of spirulina platensis has positive effect on heat tolerance of broiler embryo during last days of incubation. Also, we would like to recommend design nutrigenomics studies to clarify the spirulina substances mechanisms in the poultry embryo. At the end, I want to acknowledge and thank the Vice President at East Azerbaijan Agricultural Research Center and University of Tehran for supporting this research study. Thank you so much for your attention. And if there is any question, I am here, and please feel free to ask me. Thank you so much again.
Thank you for that insightful talk, Doctor. Now the stage is open for questions.
May I have a question?
Yeah.
Well, it's a very simple question. You have been talking about Japanese quail, and I just would like to ask that what type of quails are they were a meat type or a layer type quails?
Meat type.
Meat type. Okay. Thank you very much.
You're welcome.
I have one question. The Japanese quail hatchability was increased by the treatment by 11%, I think. What's the control hatchability in the study?
Excuse me, can you repeat the question? I didn't got it.
Of the Japanese quail, what is usually the hatchability of the Japanese quail? You said your treatment increased the hatchability by 11%.
Yeah, around 10%.
What is the normal hatchability of the Japanese quail?
Around 80%.
Thank you.
You're welcome.
Thank you for answering the questions.